Welcome to Straight Science. Straight Science is an evening science presentation series put on by University of Alaska Fairbanks Northwest Campus here in Nome, and also University of Alaska Fairbanks Alaska Sea Grant, also in the Nome office tonight. So Alaska Sea Grant and Northwest Campus, we are public servants of the Bering Strait region. We serve all peoples of the Bering Strait region, which is the homeland and waters of the Inupiaq, St. Lawrence Island Yupik, and Central Yupik peoples. Now, tonight we have Andy Ramey and Christina Ahlstrom. They are research biologists with the U.S. Geological Survey. Think about that when what the topic is, but they'll explain themselves on why the U.S. Geological Survey is handling avian influenza in all its magic and glory. And they are both with the Alaska Science Center in Anchorage, Alaska. So Christina, this is her new first Straight Science. So welcome to Straight Science, Christina. Know that, do you guys want questions at the end or throughout? If people have a question,
maybe at the end. Is that good for you, Christina?
Yeah. All right. Wonderful. All right. Hold your questions to the end. However, if you are a caller, and we will get callers have priority, and they can just ask a question whenever. It's hard to be a caller on a Zoom call, especially in our region, the Bering Strait region. You can't raise your hand or show your face or anything, do anything tricky. You just have to ask your question straight up, and callers, we appreciate that. Andy Ramey and Christina are working on avian influenza, and I was looking back in Straight Science, and Andy Ramey and Rob Kaler, management and research side of birds, they did a co-presentation, and it was just back when it was sort of like, well, maybe you might see avian influenza. That was in 2022. We were maybe just starting to get reports out of White Mountain of a few geese and things like that. And as 2022 unfolded, if you live in the Bering Strait region, we saw avian influenza, birds that were compromised and seizing or dead, and all different kinds of species. And then Andy Ramey came back, and he came back and gave us an update in 2023. And here he is again. I think he'd like to stop coming because that would mean the end of avian influenza, but he's back again. So that's good news for us, and sort of maybe we'll hear some good news. I don't know. And we're just glad Andy and Christina are taking time out to give us in the Bering Strait region an update on avian influenza. And take it away, Andy and Christina. We're so glad you're here. Thank you so much.
Thank you, Gay. Good evening, everybody. Christina and I really appreciate the opportunity to speak with you all as part of the Straight Science series. And tonight we'd like to provide some updates on highly pathogenic avian influenza, or HPAI, sometimes also called bird flu. So we're going to focus our presentation on recent detections in wild birds and wild mammals, and particularly those in the United States and here in Alaska. So we'll also provide some information on HPAI in other hosts and regions, though that's primarily going to be for context. So here's an outline for this webinar. We'll start with some introductions. Next, I'd like to provide some general background information about HPAI. Christina will then present results from recent research on HPAI in Alaska. I'll then convey some ideas on 3 things I think that we can all do about HPAI. And then we'll conclude tonight with any questions that you care to pose to Christina and I. So as we get started, we thought you first might appreciate some perspective as to where this information is coming from. So therefore, we'd like to take the next few minutes to briefly introduce the USGS Alaska Science Center, the Molecular Ecology Lab in which both Christina and I work, as well as ourselves. So the USGS Alaska Science Center is a federal office within the U.S. Department of the Interior. At the USGS Alaska Science Center, we strive to provide objective and rigorous scientific data that resource managers, policymakers, and you, the public, can use to make informed decisions regarding natural resources, natural hazards, and ecosystems in Alaska and adjacent regions. So the Alaska Science Center is not a management organization, and we do not make any policy or regulatory decisions. Christina and I both work within the Molecular Ecology Lab at the Alaska Science Center. The Molecular Ecology Lab provides genetic and genomic information on the health and status of biological resources. So stated another way, we use information from DNA, or in the case of HPAI viruses, we use information from RNA to conduct research projects designed to inform decisions made by partners. Our partners include other agencies within the Department of Interior, as well as local, state, and federal organizations. Research conducted in the Molecular Ecology Lab includes projects pertaining to wildlife health and disease, population genetics, and environmental DNA. So now I'd like to introduce myself. I was born and raised in the Midwestern United States, though I've now spent most of my life here in Alaska. For the last 20 plus years, I have lived and worked on traditional Dena'ina lands in Anchorage. I have dedicated my entire academic and professional life towards better understanding the health and status of our fish and wildlife resources. So this includes a degree in fish and wildlife biology from Colorado State University, a PhD in veterinary and biomedical science from the University of Georgia, experience working for the U.S. Fish and Wildlife Service at Togiak National Wildlife Refuge, and more than 2 decades working on research at the USGS Alaska Science Center. So at the USGS Alaska Science Center, I've been working on research to better understand avian influenza in wildlife for more than 15 years now, primarily using genetic methods. Outside of work, I spend much of my time outside. I'm an avid hiker, skier, photographer, bird hunter, and fisherman. And so now, Christina, would you please introduce yourself?
Sure. Thanks, Andy. And thanks everyone for joining us this evening. I grew up in a small town in upstate New York, where from an early age, I was interested in everything outdoors, including fishing. I traveled around for college and became interested in molecular epidemiology. This is the study of how diseases are spread by using genetic tools. And this photo was taken about 20 years ago now, when I used genetic tools to understand how bacteria were spreading among dairy cows. And this is really where I fell in love with this type of research. And I've been at the USGS Alaska Science Center since 2017, where I continue to use these methods to understand disease spread, mainly in wild birds. And outside of work, I spend a lot of time outside, like Andy, hiking and skiing and foraging for berries and mushrooms and for fishing. Yeah, that's a brief background into me. I'll turn it back over to Andy.
Perfect. Thank you, Christina. So now I'd like to begin to provide some background information on HPAI. So I'll start by conveying what I believe to be 3 particularly important points. So first, HPAI, or bird flu, is a disease caused by an influenza virus that often results in serious illness or death among affected birds. Second, HPAI was historically a poultry disease, but has recently become an important wildlife disease. Third, HPAI sometimes affects other species that have contact with infected birds. So I'd now like to provide some more information about these 3 important points. So for those that regularly attend this lecture series, you might remember some of these slides from prior Strait science presentations, as Gay recalled. So this very busy slide depicts the historical ecology of influenza viruses in wild birds and spillover hosts. I'm not going to cover all this information, but I would like to step through a few portions of the slide to describe where HPAI comes from. So not all influenza viruses affecting birds cause HPAI. There are many types of avian influenza that naturally occur among waterfowl, shorebirds, and gulls, and that do not cause disease. Birds usually appear perfectly healthy when infected with these types of avian influenza viruses. Researchers have repeatedly found these types of avian influenza viruses in wild birds throughout Alaska. This includes the detection of influenza viruses among healthy waterfowl, shorebirds, gulls, and murres inhabiting the Bering Strait region. These types of avian influenza are considered normal. Avian influenza viruses that naturally occur in wild birds sometimes spread to domestic poultry. Usually, poultry infected by these viruses also appear to be healthy. it's only a small proportion of avian influenza viruses that make poultry sick. This small proportion of viruses that cause severe disease and death among chickens and turkeys are HPAI viruses. The tendency for influenza viruses to cause serious disease and death among birds or to become highly pathogenic typically only occurs after viruses from wild birds have been introduced into poultry. HPAI viruses that evolve in poultry are readily spread amongst chickens and turkeys. These HPAI viruses may result in the death and destruction of very large numbers of domestic birds and are therefore very costly to the poultry industry. The spread of HPAI from poultry to wild birds, depicted here with a red arrow, has only become common since 2002. So I'd now like to show a few maps to convey how outbreaks of HPAI in wild birds have become more common and more geographically widespread. So this map depicts detections of HPAI among wild birds over a span of approximately 4 decades, more specifically from 1961 through 2002. During this period, HPAI was detected among wild birds exactly twice. The first detection was made from terns found dead in South Africa in 1961. Approximately 4 decades later, HPAI was again detected in wild birds, this time in a park in Hong Kong in 2002. During 2005 to 2009, outbreaks of HPAI affected wild birds throughout much of Europe, as well as areas of Asia and Africa, as depicted on the map on the left side of this slide. During the next 10 plus years, HPAI became more widespread within these regions, as depicted in the map in the middle of this slide. Wild birds inhabiting North America were affected by HPAI for the first time in recorded history during the same time period. Since 2021, HPAI has become even further geographically widespread, having now affected wild birds inhabiting most continents, as illustrated on the map on the right side of the slide. In recent months, HPAI has spread to wild birds inhabiting Antarctica for the first time. Australia currently represents the only major continental region at which wild birds remain unaffected by HPAI. Within the United States, the USDA has confirmed HPAI among wild birds sampled in all states except for Hawaii. Types of wild birds commonly affected by HPAI in the United States have included ducks, swans, geese, eagles, hawks, owls, ravens, gulls, and terns. Some of these HPAI detections were made from samples collected from apparently healthy birds. This has been particularly true for samples collected from ducks. Many more detections of HPAI have been made using samples collected from relatively fresh, representative carcasses found as part of mortality events. This has more often been the case for detections among raptors, seabirds, and ravens. So in the prior slide, I mentioned mortality events. Mortality events occur when numerous animals die during a relatively short time interval and at the same general location. Mortality events sometimes involve large numbers of animals and may represent a conservation challenge, particularly when they involved a threatened or endangered species. For example, during 2022, an estimated 1,500 threatened or endangered Caspian terns are estimated to have died from HPAI as part of a mortality event occurring in Michigan and Wisconsin. Around the same time in 2022, another example of a significant mortality event occurred in eastern Canada. In this mortality event, it was estimated that at least 40,000 seabirds died on account of HPAI, with northern gannets being the species affected in highest numbers. Mortality events sometimes involve diverse types of animals, such as both wild birds and mammals. For example, a mortality event in Puget Sound in Western Washington during 2023 involved the mortality of approximately 1,700 wild birds, mostly terns, as well as numerous harbor seals. For context, the spillover of influenza viruses from wild birds to marine mammals is not entirely new. For decades, there have been sporadic and isolated outbreak events caused by the spillover of influenza viruses from wild birds to marine mammals. However, marine mammal mortality events associated with influenza viruses have become increasingly common within the past several years. For example, HPAI has recently been associated with several particularly large marine mammal mortality events in South America. Recent reports from Brazil, Peru, Chile, and Argentina have estimated the loss of hundreds to more than 10,000 seals and sea lions as part of mortality events attributed to HPAI. Here in North America, a relatively large marine mammal mortality event on the east coast of the United States during 2022 was associated with HPAI. It was estimated that approximately 330 harbor and gray seals died. Generally, HPAI-associated marine mammal mortality in North America has, so far, been reported to be of smaller magnitude as compared to reports from South America. In Alaska, there has been only a single detection of HPAI in a marine mammal. HPAI was detected using tissue collected from a polar bear carcass found near Ukakvik in 2023. To date, HPAI has not been detected among seals or sea lions inhabiting Alaska. The spread of HPAI from wild birds to mammals is not limited to marine species. This map, generated by the USDA, depicts all detections of HPAI in mammals within the United States. By using the key on the right side of your screen, you can see that numerous types of terrestrial mammals have been affected by HPAI in the US, especially carnivores such as large cats, bears, foxes, skunks, and raccoons. Here in Alaska, mammal species affected by HPAI have included black bear, brown bear, polar bear, as mentioned in the prior slide, and red fox. So speaking of the spread of HPAI to mammals, there's been a lot of concern voiced in recent years about potential spread of HPAI from wildlife to humans. So I want to provide context for this concern by showing you data reported by the CDC. The CDC is the lead agency in the US when it comes to HPAI and human health. So this plot, taken from the CDC website, depicts the geographic and temporal distribution of human cases of HPAI reported globally since 1997. The vertical bars represent the number and geographic origin of human cases reported in each year. As you can see, most human cases have been reported from North Africa and East or Southeast Asia. The vast majority of these human cases have been associated with exposure to infected domestic poultry. Human cases of HPAI have become less common since approximately 2015, despite HPAI becoming increasingly common among wildlife. Here's a screenshot from the CDC website taken this morning. To date, there has been only one reported human case of HPAI in the US. The single affected person had extensive exposure to infected domestic poultry and fully recovered after reporting fatigue. As of today, 28 March, the CDC reported the public health risk associated with HPAI to be low on their website. So this slide helps to summarize the background information presented tonight by describing the current ecology of HPAI viruses in wild birds and spillover hosts in Alaska. Recent HPAI viruses have evolved such that they are now readily maintained and dispersed by wild birds, largely independent of poultry. This has facilitated their rapid geographic expansion. HPAI viruses sometimes spread from wild birds back to domestic poultry in Alaska. HPAI viruses have also sporadically spilled over into wild mammals, including foxes and bears. These HPAI viruses have caused some level of mortality among virtually all affected animal hosts in which they've been detected. So at this point, I'm now going to turn the presentation over to Christina so that she can convey some of what we've learned through recent research on HPAI in Alaska using genetic tools.
Thanks, Andy. Scientists at the Alaska Science Center have been researching avian influenza viruses for a long time. For the past 15 years or so, we've been collecting about 1,000 samples from mainly hunter-harvested waterfowl as well as some environmental fecal samples in areas around Eisenbeck National Wildlife Refuge in the Aleutians East Borough. Why this region? It's an area where many species of wild birds congregate during migration from multiple flyways. It's also a common area of sport and subsistence harvest of waterfowl. And through genetic analysis of those low pathogenic avian influenza viruses that Andy introduced, we found that this is a region where viruses are routinely introduced into North America from East Asia. And until 2022, we only detected low pathogenic avian influenza in this region. Then, in 2022, we did detect HPAI in several samples from this region, and genetic analysis indicated that there were multiple types or different genotypes of these viruses in these samples. And this suggested that HPAI was introduced into this region multiple times. And when we compare the genetic sequences of the viruses to reference databases, we find that 2 were most closely related to viruses circulating in Asia, and another was more similar to viruses detected elsewhere in North America during this ongoing outbreak. So we published this research last year, and here's a link if you'd like to read more. I think it's important to mention that high-path avian influenza is different from low-path avian influenza, given its disease severity and economic importance. Only one lab in the entire country can confirm if samples are high-path. So when a wild bird is suspected of HPAI in Alaska, samples move through this pipeline to the USDA confirmatory lab. And this is also the case with backyard and commercial poultry and wild mammals. They're all fed through the USDA lab, including samples from all other US states. So unsurprisingly, we have seen some delays in reporting, but we were fortunate to work with folks at USDA to obtain the genetic sequences of all confirmed high-path avian influenza viruses detected in Alaska in 2022. And these included 14 viruses from backyard poultry, 159 from wild birds, representing 24 different species, and 4 viruses from mammals. And as Andy mentioned, this included a black bear, a brown bear, and 2 red foxes. And these samples came from 16 Alaska boroughs or census areas. And here, well, it's a large file, so it takes a minute to change. Here's a map indicating where samples were obtained. Boroughs with 5 or more virus detections are colored, and those with 1 to 4 detections are shaded in gray. The highest number of samples came from the Aleutians East, which is largely driven by the targeted surveillance efforts I described, followed by the Matanuska-Susitna Borough and the North Slope. And on the left-hand side of the screen, you can see the species from which viruses were obtained, the number of positive samples from each species, and the location where they were detected, and that's indicated by colored circles. And the highest number of samples were from northern pintails from 5 boroughs. The next highest number of samples were from mallards, followed by bald eagles. And asterisks indicate species were detected in the Bering Strait region, so either from the Nome census area or the Northwest Arctic Borough. And these included glaucous gulls, a common raven, and a red fox. And here are the number of samples collected over time. This is all through 2022. There are a lot of colors here, which is a point I'd like to convey. Samples were collected from geographically diverse locations throughout the year. It's also worth noting that 2/3 of the samples were collected in the last quarter of the year, from September through December. And this may be partly a function of increased surveillance during this time. And asterisks indicate the samples collected from the Bering Strait region. There are 4 asterisks shown. 1 sample from Nome didn't have or had an unspecified date, so it's not shown here. And as I mentioned, I'm really interested in understanding how diseases spread by using genetic tools. So here we were able to use the genetic sequence of the viruses to understand how high-path avian influenza moved through space and time and in different hosts in Alaska. We detected 7 different types of high-path viruses, which I call different genotypes. And we found a pattern where 4 of these genotypes showed relatively limited spread and 3 were more widespread. And these different letters, to those of you who can see the slides, and numbers indicate the names of these different types of HPAI viruses detected in Alaska. It's the USDA naming convention. And in the Bering Strait region, 2 of these genotypes were detected, 2 of the more widespread genotypes. But I'll walk through all of the genotypes detected in Alaska. Genotype A4 was detected in 6 wild birds, all from the Aleutian Sea sparrow. And when we compared the sequences of the viruses to reference databases, this is one of the ones that most closely matched viruses circulating in Japan. So this was likely an introduction from Asia. And the 4-letter codes at the bottom of the screen, they're abbreviations for different wild bird species. So you can get a sense of how many different species were affected. And in this case, it was 4 different species of waterfowl. Genotype B2.1 was only found in the borough of Anchorage in a bald eagle, though this genotype was frequently detected elsewhere in North America. The minor genotype was only detected in a northern pintail from the Aleutians East borough. It was not detected outside of Alaska, which is why it wasn't assigned a letter and number per the USDA naming convention. Genotype 3.1 was detected in 5 wild birds in 4 different Alaska boroughs, but only over about a 2-week period from the end of May to early June. And now I'll describe the more widespread genotypes. B4.1 was detected in 18 wild birds, 1 mammal, a black bear from the borough of Juneau, and 3 backyard poultry. These samples were collected from 10 different boroughs, and it was also detected in 3 North American flyways. Genotype A3 was detected in 26 wild birds and 1 red fox from the Aleutians West census area. It was detected in 6 boroughs, including in 2 glaucous gulls from the Nome census area. And this is the other genotype that was most closely related to viruses from Japan and was likely introduced from East Asia. And unlike the other virus genotype introduced from Asia, this one spread outside of Alaska into the lower 48 in Canada. Lastly, genotype B3.2 was the most commonly detected genotype in Alaska. It affected 72 wild birds, including a common raven from the Northwest Arctic borough and a glaucous gull from the Nome census area, as well as 1 mammal, a red fox also from the Nome census area, and then 5 backyard poultry from elsewhere in the state. And this genotype has been detected widely outside of Alaska and has caused a number of die-offs, both in seabirds and marine mammals. And here I'm summarizing the number of samples from each genotype detected in Alaska. Those first 3 are the more widespread genotypes. We were interested in understanding why we might see differences in spread among these different viruses. One explanation could be differences in pathogenicity or transmissibility. However, this is not really supported by the data elsewhere outside of the state of Alaska, because, for example, genotype B2.1 was detected in only 1 sample in Alaska, but it was detected widely in the lower 48. So another explanation could be the timing of introduction of these different viruses into the state. We can use the genetic data to estimate when each was introduced, and this is indicated by the months at the bottom of the slide. And interestingly, the 3 most widespread genotypes were also the 3 estimated to be the first to be introduced into the state. While additional data would be needed to confirm if timing of introduction affected virus spread, it is logical if we think about the immune status of wildlife. So at the beginning of an outbreak, most animals have had no prior exposure to the virus and are susceptible to infection, and this would be indicated by the blue line. As time goes on, some animals survive and develop antibodies that prevent them from becoming infected. So it's possible that by the time the last 4 viral genotypes were introduced into Alaska, there was enough population immunity that these viruses were not able to take hold and spread. So now I'll summarize some of the recent research we've been doing at the USGS Alaska Science Center on high-path avian influenza. We identified numerous virus introductions into the state from multiple flyways between approximately November of 2021 through August of 2022. We find extensive transmission among species, including mammals. We found 2 of the more widespread genotypes in the Bering Strait region, and spread differed among genotypes, likely driven by timing of introductions. And through collaboration with lots of partners, we hope to continue monitoring and genetically comparing avian influenza viruses to better understand their evolution and dispersal. And with that, I'll turn it back over to Andy.
Thanks, Christina. That was outstanding. So now I'd like to speak about a few things that I think we can all do about HPAI, should there be future detections in the Bering Strait region or anywhere else in Alaska for that matter. So I want to start by being clear and upfront that there's really nothing that you, nor I, nor that anyone can do to stop the occurrence and spread of HPAI among wildlife at this point in time. However, I think we can all choose to be informed, to be prepared, and to take appropriate action if there are additional detections of HPAI in Alaska, or if we witness wildlife that we suspect to be affected. So I'd now like to talk for a few minutes about each of these bullet points. So the first thing we can all do about HPAI is to be informed. So you're doing this right now. By attending this webinar and seeking additional information about HPAI, you're going to be better able to make informed choices about your harvest, cleaning, and cooking practices based on your own perception of risk. You will also be able to make more informed choices about how you will respond if you witness wildlife that you believe to be affected by HPAI. So how can you determine if a bird or other wildlife is affected by HPAI? The reality is that you can never be certain whether a bird or any other animal is affected by HPAI without laboratory confirmation. Even then, we have to recognize that laboratory tests have limitations and involve some degree of uncertainty. Also, some types of birds, and particularly ducks, often appear healthy or asymptomatic when infected with HPAI viruses. Therefore, it may be difficult or even impossible to be absolutely sure if animals you observe are affected by HPAI. That stated, many animals affected by HPAI become sick and may die. Therefore, animals that appear to be obviously sick, those that do not try to escape when approached, or those that have died from an unknown cause should generally be considered suspect. Common signs of HPAI among sick birds include a lack of coordination, stumbling, inability to stand upright, inability to fly, swimming in circles, a twisted neck, or paralysis. So now let's take a look at a few examples of these signs. So at this point, I'd like to play several short videos with examples of behaviors that have been associated with HPAI in wildlife. So these behaviors do not confirm that an animal is affected by HPAI, but they are extremely suspect in the types of behaviors that I encourage you to look for and to report. So if you prefer not to see short videos of sick animals for the purpose of better understanding what you might encounter in the field, I encourage you to please briefly turn off your monitor or look away from your screen for the next few minutes, and I'll let you know when 4 short videos are over. So now please bear with me here while I try to navigate multiple screens here and get some videos to play. So this first video was made in the Bering Strait region near Golovin in May 2022 and was shared on the Leo network by Hunter Gray and Gay Sheffield. Swimming in circles, as displayed by the Brant goose in this video, has repeatedly been associated with HPAI affected birds. So this next video is of a bald eagle in the Seattle area and was posted to YouTube by Kirsty Mule, an urban conservation specialist. So this eagle had poor coordination and wasn't able to fly. Again, these are commonly reported signs for HPAI affected birds. The third video I'd like to show is of a sick bald eagle that was posted to Facebook by Who's Woods Raptor Center. The eagle in this video was encountered in Iowa and was ultimately confirmed to be affected by HPAI. This eagle displayed a twisted neck and was readily approached. Again, these signs are suggestive of HPAI infection. So now let's look at a video posted on YouTube of 6 sea lions from Argentina that are suspected to have died from HPAI. Signs of HPAI among affected mammals are less well described as compared to birds, but generally have been reported to be neurologic. Animals may display a lack of coordination, exhibit tremors, and be readily approached. So for anyone that had turned off their monitor or has been looking away from the presentation, I encourage you to please turn your attention back to slides as the videos of sick animals are now over. So I'd like to continue with just a few more slides pertaining to being informed. So to obtain additional information about recent HPAI detections in wildlife, you can find a list of all USDA confirmed laboratory detections among wild birds and wild mammals sampled within the US at the websites indicated here. Links to these websites will be posted in the chat. To become more informed regarding human health and safety on account of HPAI, please consult the CDC website listed at the bottom of this slide. The link to this website will also be provided in the chat. If you are a hunter or help to process wildlife that has been harvested, the USDA and US Fish and Wildlife have issued guidance regarding steps that can be taken to reduce the risk of HPAI to human and domestic animal health. Suggestions offered by these agencies include disinfecting knives, equipment, and work surfaces that were in contact with wildlife during butchering, as well as cooking meat to an internal temperature of 165 degrees Fahrenheit. Links to the USDA and US Fish and Wildlife Service guidance for hunters can be found at the websites indicated here, which will also be posted into the chat. So now that you are becoming informed about HPAI in wildlife, the next thing you can do is to be prepared. So I encourage you to develop a personal response plan to help guide what you will do if there are additional detections of HPAI in your region, or even if there are indications that unusual mortality among wildlife could be on account of HPAI. So to clarify, I'm not proposing that you develop a detailed response plan or follow a manual like the documents used by national and international organizations as depicted on the left side of the slide. Rather, I'm suggesting that you consider taking some time to think through your options for modifying some of your current practices as represented by images on the right. For example, is there anything about your practices for butchering or preparing harvested animals that you think may help to lower your risk of being exposed to HPAI? Have you carefully considered your practices for washing your hands, your knives, and work surfaces, and even your rubber boots following the harvest and processing of animals? What will you choose to do if you observe sick and dead wildlife? Per the latter, I encourage everyone to consider reporting their important observations of sick or dead animals. Your observations may help to raise situational awareness in your community and guide organizational response. When reporting sick and dead wildlife, it is extremely helpful to include the date and time, the specific location, the species affected, and the number of animals that appear sick and that have died. Taking photos or videos with your phone can be extremely helpful for documenting what you observe. Speaking of photos, please consider taking a photo of this slide and adding these reporting numbers to your contacts. To report sick or dead wild birds, please call the U.S. Fish and Wildlife Service Alaska Sick and Dead Bird Hotline at 1-866-527-3358. Observations may also be reported to the LEO network, which can be accessed at the site listed here. To report sick or dead,
I'm gonna
please.
just, this is Gay. I'm just gonna ask, when it matters out here, to report sick or dead wild mammals, you're talking about land animals in this case for the state of Alaska. Is that correct? Not marine mammals?
The guidance that I've been giving is all mammals to call fish and game.
Thank you.
That's the guidance that I got. I realize that NOAA does lots of work with marine mammals. Per the state of Alaska, they encourage you to call your Alaska Department of Fish and Game office. You can contact Fish and Game at the email address listed here, or by calling 907-328-8354. All of these contacts will be posted in the chat. And if you have problems reporting your observations from the Bering Strait region, please contact Gay Sheffield. Gay has been helping with HPAI response since these latest outbreaks began and is an absolute champion for your region. So once you are satisfied that you have become informed about HPAI and you have prepared your personal plan for response, you will be ready to take appropriate action. I encourage you to use information about HPAI from credible sources, such as those mentioned tonight, to make informed decisions about your harvest, cleaning, cooking, and reporting practices. This may be especially important if there are additional detections of HPAI in your region, or if there are indications that unusual wildlife mortality in your area may be on account of HPAI. Also, please remember that reporting your observations of sick and dead wildlife may ultimately help others in your community, your region, and throughout Alaska to make more informed decisions. Thank you for your time and attention, and Christina and I will now be happy to answer any questions that you have for us.
Awesome. Thank you so much, Andy and Christina. And while people are thinking of their questions, I see there's one in the chat already, but while you're thinking of raising your hand and whatnot, this is the time for Strait Science audience to show a little love in the chat for Christina and Andy. It is never easy to be a public speaker, and we thank them so much for making time after hours in their work week to contact us and let us all know a little bit more about avian influenza. So throw some love their way in the chat, or some people, look how good some people, there's Julie Matt with you right there. She's happy. So that's really good. I know I have a lot of questions, and we'll throw this open to the group, and just thanks again for giving us an opportunity to hear. You're the only ones that come back to talk about this avian influenza, and it's something that we're living with. So with that, let's see, there was a question in the chat, lots of good info. Few questions. Does highly pathogenic mean the same as highly contagious? That's first question. Julie, you went heavy on questions. Can you re-explain how samples are tested and moved through confirmation of HPAI, the highly pathogenic avian influenza? There was not a lot of Kodiak data, nor is there a lot of Bering Strait data, I have to say. Was there a positive on a Kodiak eagle in spring 2023? I don't know if you can hit all these at once, but
I'll take them one at a time.
okay.
So highly pathogenic, that's a great question, and the answer is a little bit nerdy. So highly pathogenic has a very specific technical definition, and it has to do with a virus's ability to kill chickens. So there's specific tests that chickens are inoculated with a virus to determine exactly how pathogenic they are. It is not the same as contagious. There can be naturally occurring flu viruses that can easily spread that don't cause any disease. Those are very contagious. They don't cause disease. So highly pathogenic is the ability to cause disease, and specifically in chickens.
Cause disease and death?
In chickens, yes.
Okay.
How samples are tested. So the traditional pipeline per a slide that Christina showed would be that a sample is tested for a general influenza virus, usually through a genetic test. Is there a marker for influenza virus? If it is positive on the marker for an influenza virus, then it goes through another marker for potential highly pathogenic flu virus. If it has that marker for a potential high pathogenic flu virus, then it goes to the national laboratory, and there they try genomic sequencing. If the sequence for that virus matches high pathogenicity, it's confirmed. If they get, that is the gold standard. That's how they know it's confirmed for high path. And so, for example, those were all confirmed confirmations that Christina described through the sequencing of, and then the analysis, 177 full genomes from Alaska. Those all had genomic sequence for the entire virus or most of the virus. It's one particular gene that determines if it's highly pathogenic.
Okay. I've learned tons in this 1 hour or less than an hour period. I tend to clump avian influenza. I don't know very much about it. I asked that question about the testing because I'm not sure what I saw, but we had an eagle demonstrating some severe distress with swinging of the neck. And I study harmful algal blooms. So I was looking at that thinking, is that eagle sick from HABs? And I actually sent the video to Gay, and she said it looks like avian influenza, which honestly didn't cross my mind until she said that. It took a long time for that eagle to be removed. It was in a very, very public location in Kodiak. And so I have a lot of questions around that particular incident. And I tried to follow up on it. I never really heard. I kind of heard from backwater that it was sick and euthanized, and it likely had influenza and died. Some of my questions, and you can take them in order after other people's, is that it was really in a public area, and my dog was trooping back and forth in front of it too. A lot of off-leash dogs, a lot of people. It was snowy and muddy. The eagle was sitting in all of that. And what is the likelihood of all of us tracking some kind of nasty virus around or having an animal pick it up, like our pets, if it was contagious? It took a long time for our response to get it into effect. It was probably out there public for at least 4 or 5 hours before somebody took it.
So I can tell you, we've conducted some experiments. There's a good bit of literature that influenza viruses can remain infectious in the environment for a very long period of time in cold water. So cold, fresh water is a generally happy place for a flu virus to remain infectious for long periods of time. What is the risk? I can't tell you. I don't think anybody can put a number on that. It's sort of, what your perception is based on how many birds are there, what the temperature is, what the salinity of the water is, a lot of different things. But this is exactly the sort of case where I encourage you and others to think about. We know high path influenza is on the landscape nowadays. We know it can be spread among different species. So I think it's good to be thinking through these sorts of scenarios that you can consider what you're comfortable with. There's great recommendations for how you can kill flu viruses with heat, with solutions, with bleach, washing off rubber boots. So I think it's good things to be thinking about so that when you see something that you think isn't right, and if you've been walking in that area, that you can take make the appropriate response that's good for you to help mitigate that risk, so that you feel confident that you've done what you can to mitigate the risk for that virus to be spread via your boots or your actions.
All right. I think that covered it all. Is that right? Thanks, Julie. And thank
Great
you, Andy.
questions.
We've got several callers on the line, and I just want to reiterate that callers can unmute. I think it's pound or *6 can unmute. If you cannot unmute, and we have had this happen on several Straight Sciences, just text the old cell phone here and I can read them out. And my number is 907-434-1149. So if you're in one of our more remote areas and the mute isn't working for you, then just call and we'll ask the questions and get you your answer. Let's see. There is a question from Sarah Schwartz. I may have missed the info earlier. Is this always fatal, or are there any cases of recovery for any animals and birds? If so, is there any information on treatments used? If, for instance, a chicken owner has if a chicken owner has a bird death due to suspected or confirmed avian influenza, what is the process for disposal of the carcasses? And do note that here in the Bering Strait region, we do have several entities that have home chickens.
Christina, I don't want to do all the talking here. Do you want to handle this, or?
Go ahead, Andy. You're doing a great job. If there are more specific questions on genotypes, I'd be happy to chime in.
Okay, so HPAI is not always fatal. We know that some species recover, and we don't have full information, honestly, about what's going on in all species at the moment. Things are changing so fast. This HPAI is, I showed those maps of spread, is breaking all the rules these days, and so lots of species are becoming infected that haven't been infected in the past. But we do know through testing, through serology, Christina talked about recovered animals, that some animals are recovering. So no, this is not always fatal. I think it depends a lot on if animals have seen influenza viruses in the past. We talked about all these naturally occurring viruses that are in the environment. Those might actually be a really good thing for lots of animals. If birds see these low pathogenic viruses, that might help to protect them against serious illness and death. So there can absolutely be non-lethal infections. If a chicken owner has a death that is suspected or confirmed for avian influenza, I encourage you strongly to contact the state vet at ADEC and to get your guidance directly from Dr. Sarah Coburn and her shop. I think the processes, they have changed during this outbreak as far as quarantine procedures for premises. I'm not certain that I know what the latest is with all the changes that have occurred, but Dr. Sarah Coburn will know exactly what they are. So reach out to ADEC, the Alaska Department of Environmental Conservation, the state vet. Sarah and her office will tell you exactly what to do in that case.
And if you're a gnome chicken owner or pet bird owner and you suspect there's a problem, you go straight on down to ADF&G's regional office here on Front Street and let the ladies or men at the desk know that you have this situation, and they will make all the phone calls and get you who you need to talk to. So that is what I would do in the region. If you are a poultry person, and we have several people that are for lots of reasons raising their own birds, given the price of eggs and birds out here. In Nome, we have several poultry owners. So go down to Fish and Game and they will get right on it. Looks like there's a question from Brianne Rogers. Looks like pintail often test positive, but aren't having big die-offs. Theories on why? Question.
And it's another great question. I think this has to do with those naturally occurring low pathogenic viruses that don't cause disease. We have been studying those sorts of viruses in pintails for 15 plus years in Alaska now, and we know that pintails very commonly harbor those viruses. And maybe it's sort of ironic, but that's probably what's going for them right now, is that they are being protected against maybe not infection, but against disease and death by the fact that they've been exposed to these low pathogenic viruses. So that is my best guess on what's going on there. And I think that's true not just of pintails, but of many species of ducks. I think we're also seeing that with mallards. We're seeing that with teal, with widgeon, that the ducks, many of the ducks that are coming back HPAI positive seem perfectly healthy. I think they're able to cope with the infection. Not always the case, but it's very often the case.
All right. Thank you so much. I see a lot of the island on as callers and attendants tonight. Does anyone from St. Lawrence Island or outside of Nome in the Bering Strait region have a question for Christina or Andy regarding avian influenza in our birds, seabirds, eggs, marine mammals, land animals? I mean, this stuff seems to be transmorgifying into anything it seems to be able to jump quite rapidly. Any questions? And feel free as well, callers, to jump on at any time. Sue Moore has a question. Great presentation. Were there any transfers from birds to marine mammals reported for the Bering Strait region or the North Slope Borough? The fox and the bear?
So to marine mammals. So
Oh, I guess yeah, the fox.
specifically to marine mammals, I would say using data from 2022, the data that Christina presented, we did not have any marine mammals. We do not have the data, the genetic data from 2023, but there has, the only marine mammal that's been reported in Alaska so far has been a polar bear. So we don't have the ability to conduct analysis to determine the origins from that polar bear. So I would say we don't have information to assess marine mammals in Alaska, but I do want to clarify the only detection in Alaska to date in a marine mammal has been that single polar bear sample, at least to my knowledge.
And my question then would be, and you can't answer it, is what is the effort sampling marine mammals and testing them for avian influenza in marine mammals? And I can't tell you what that is at this point either.
Alaska is a big place, and that's why your reports are so helpful for understanding what's going on. So I just want to thank anybody and everybody that is on the call, that has made a report, that has helped with testing. I see there's some hunters on that have really allowed us to sample their hunter harvested birds. That is so helpful for understanding what the heck's going on. So thank you all.
Jeff Wassley says, great info, Christina and Andy. Are most of the mammals getting the virus from scavenging sick or dead birds, or are some contracting the virus from the environment? Good question.
That's an excellent question. And I think we don't fully know, but I think there are certainly anecdotes to suggest that many mammals are picking it up through scavenging. I think there's also some head scratching cases when we look at cases or events such as some of those that have occurred down in South America, where it's hundreds or thousands, or in one case, more than 10,000 animals that have been reported to be sick from HPAI. That seems to me less likely from scavenging than an environmental source or even something else. So I don't know, honestly. I think that's an outstanding question. I think that's a question that a lot of people are scratching their heads and trying to get to the bottom of. Outstanding question.
All right. And that takes us currently through the chats. So if the callers have questions, I don't see anyone raising their hand. I have a couple questions at the end but I guess I'll get in right now. Are we going to live with avian influenza now? It's the genie seems to be out of the bottle. In 2009, there was a big effort by USDA to look for it. Remember, we were pretty familiar in this region with 1-800, call 1-800 bird flu. And they came out and did a lot of testing in this region and didn't find it up here. But now we have it. And whether or not the response is adequate, that's a whole nother question. But are we going to be living with this forever now? Or does it just slowly, we build up those antibodies? Like there's birds that have recovered from it. Maybe there's marine mammals that have recovered. Maybe the foxes recover from it. Bears, I don't know. But we're getting into things that are a little nerve-wracking if your livelihood is accessing clean, healthy populations of many, many different food sources in this region. So are we just going to have to learn to live with it? And if that's the case, will there be some consistent monitoring and surveillance so we know where we are with it?
So, Gabe, no one knows the answer to this, but I want to provide
Yeah.
some historical perspective that you can either use historical perspective to be an optimist or a pessimist. So, which do you want to be first, an optimist or a pessimist?
Optimist. I want to be an optimist. Get rid of it.
So, from an optimistic side of point, if we look at, I often refer or go back to what we know about all these naturally occurring viruses because they've proven to be so helpful for understanding what the heck's going on with HPAI currently. So we know with these low pathogenic viruses that sometimes there's what's called a selective sweep, which is actually probably what's going on right now, where you get a virus that's super successful and it outcompetes the competitors and it takes over and the competitors disappear. We know that that happens with low pathogenic viruses. Again, I think that's basically what's happening right now with this high path H5 virus. So if there eventually becomes a low path H5 virus that is more transmissible, which again, we just talked about that before, that's different than pathogenic. If it's more transmissible than the current HPAI, it might spread around the world and lead to antibodies that are similar enough that HPAI dies out. We've seen similar things happen with low path viruses in the past. So there's sort of a roadmap that could happen. So there's the optimistic side. The pessimistic side, if you look at those maps that I showed going back to 1961, the first detection of wild birds, we went from 2 detections over 41 years to whatever you want to describe this show that we got going on nowadays. Things have changed quite a bit and the trajectory is frankly not good. So I don't know what the future holds, but I think there's reason you can be optimist. And there's also maybe a reason to say, maybe this is the new normal.
Buckle in. All right. I guess we just takes time to figure this out. Thank you for showing the video on the marine mammals that have the avian influenza or suspected to have it, like that sea lion on the beach and wherever. And just to reiterate, it had tremors, lack of coordination, and approachable, I would imagine. He wasn't going to, it didn't look like they were going to go anywhere. So that's really helpful for us. And I would add to, if you are concerned about what you're seeing in marine mammals, I would get a hold of, if I lived in the Bering Strait region, I would get a hold of your COERIC Natural Resources Director, Subsistence Director, Chuck Manadaluk. I would also get a hold of your co-management, marine mammal co-management representative. So whether it's the Alaska Beluga Whale Committee, the Ice Seal Committee, or the Alaska Eskimo Whaling Commission, I would get a hold of my commissioner to report, or I would call the NOAA 1-800-24/7 stranding hotline, even if it's walrus or polar bear that is, and we also have the Alaska Nanook Commission. I don't want to leave those guys out here in Nome. Eskimo Walrus Commission here in Nome. I would make sure that the representatives, there's tons of ways to communicate. There's an issue and all of those will be passed along and get an answer. You can always call me as well, but in our region, we have a lot of different resources. We're really thankful, Andy, for the ones you have listed. We also have our co-management birds as well, the Alaska Migratory Bird Co-Management Council. All of those have reps in several of our villages throughout the Bering Strait region. So I would say we have lots of ways. What we're looking for then is these tremors, lack of coordination, and sort of approachability. How do we, or there's no answer to this question either, but we are having concurrently at the same time as avian influenza may arrive, we just experienced in 22 the largest, most persistent, most toxic bloom of Alexandrium catanella nationwide ever. And so this region is being pressured extremely hard in all sorts of ways in the environment and in the ecology of the marine system. You know, and I guess people ask, how will we know? Is it harmful? It's terrible now. Is it harmful algal bloom? Is it disease? I would say whatever you see, continue to report to all the people that Andy recognized and all the ones I just mentioned, your co-management partners, COERIC, Alaska Sea Grant, any of those NOAA stranding hotlines. If calling NOME is easier, then call NOME. If calling Anchorage is easier, call Anchorage. We need now, we are on our own in the Bering Strait region. We don't have any federal biologists here for the birds or the marine mammals. And we have lots of different sort of issues. Again, I'm an optimist. We can do this. People have gotten through a lot worse in the Bering Strait region in the last 1,000 years. And so report what you see. We are an incredibly vigilant and knowledgeable region. Report what you see. And that there are people that will respond to what your questions are and concerns are. And like Andy said, use this as a weapon. Take a video, take a picture, and ask for help, ask for information. It's important to document it, and that way we may get more service out here and further north as well, if we can show, hey, you know. And we sent, just so you're aware, Andy, in 2022, the reason why there's only 4 birds that light up, or 3 birds and a mammal, we were only allowed to send in 15 birds because we weren't able to, you know, there was a scare, people don't touch the dead birds. So literally those were from 15 birds and 3 of them tested positive. If that gives you some idea of what our ratio of excitement was around here, it is, those were just 15 pristine birds.
And you raise a really good point, Gay, that when people, I hope that some people go and look online at some of these resources, look at the number of dead birds reported, where they are, the species they are, same with mammals. We know these are gross underreports. That is, the numbers reported, you know, the more than 9,000 wild birds reported on the USDA website is a drop in the bucket to what the real number is on the landscape. We know that. And we know it's not underestimated in the Bering Strait region, elsewhere in Alaska. The way that we can help to improve what's going on is through people's reports. When they call in a report and say, you know, not only did I see dead gulls, but I saw exactly 22 dead gulls, and I surveyed an area, or I looked at, you know, only part of a beach. I think there was more gulls. Then hopefully we can get some resources out there to better understand exactly how many gulls that represents. But the more complete a person's report can be, the more information we have to get a better sense of the denominator, the number of birds truly affected here. And everybody, myself included, should look at these numbers and say these confirmed detections are grossly undershooting what the true disease burden, the true number of animals that have experienced disease, that have been sick and died, really is.
Yeah. And thank you for saying that, Andy. That is straight up, and very appreciative to hear it. Oh, there seems to be some activity in the chat. Oh, everybody's just thanking you. So that's nice. And any other questions? St. Lawrence Island, this is a good time to ask if you have any questions on this avian bird flu situation, both land animals, perhaps, ocean animals, and the birds, seabirds as well? Or the callers?
The island is another prime place where this is Cheryl. I'm
Hi,
with the
Cheryl.
native village of Gamble. A lot of birds migrate through here. We get a lot of bird watchers and stuff, and it would be nice to be able to monitor the birds. Not just that, but we do a lot of hunting for birds. I just wanted to point that out. I do remember, I think several years back, where a whole bunch of birds died off, I think along the north side of the island. Gay, do you remember?
2013, is that the avian
Yeah,
cholera? That was the avian cholera event.
okay.
Yeah.
Yeah, it's, just remembering and putting forth that information. I don't remember a massive die-off like that since, but something to keep in mind.
Thank you.
Great observations. Thank you, Cheryl. I sure hope the birds of St. Lawrence Island do well this year.
Thank you. Thank you guys for coming on and giving us a whole bunch of good information. We appreciate it.
Well, you're so welcome, Cheryl. We're very appreciative of the opportunity to come talk with you all today and hear your observations and yeah, interact with you. Thank you.
Okay, I forgot my camera's off, but just to say hi and show my face. Nice
Okay,
to
thank
meet
you,
you.
Cheryl. Nice to meet
Mhm.
you.
This is Erica, also with the Native Village of Gamble. I had a
Go ahead.
question. Are there any funding to monitor birds?
Can you repeat that? I didn't catch it.
Are there any types of funding to monitor birds?
Funding to monitor birds.
So Erica,
Yes.
is your question then is regarding to monitor birds for avian influenza specifically? Is that what I'm understanding?
Yeah.
Hmm.
And is there anywhere we could send dead mammals, or how do we find out if it had died from the avian influenza?
So like, are you talking like a marine mammal or a fox or a bird or all of them?
All of them.
All of them. Okay.
So Erica, when it comes to funding for test or to sample for avian influenza, there is some funding, but it's not as lucrative as you might hope given what's going on. I think there's competing things happening. One, federal dollars and state dollars are being pinched, declining, and also this outbreak is getting more widespread geographically among more species, so the funding is also getting diluted. So speaking from the federal side of things, I'm most familiar with my agency. Right now, our funding for avian influenza for the entire country is far, far, far smaller than it was. Gay talks about back in like 2009. It's way smaller than it used to be. And so I think that is a huge factor in the level of response that's being seen, and not only in the Bering Strait, so elsewhere. So right now, there's limited funds for testing, and there's more funding for testing when people are able to report carcasses. That's probably the best way to get testing accomplished, is to use the numbers that were provided. I'm happy to provide those again if that would be helpful for wild birds. For mammals, Gay provided some great local resources. I think contacting Gay is a wonderful, Gay's such a fantastic resource for your region in order to put you in touch with the right people. That's probably the easiest way to get samples tested in the short term. Sometimes there are opportunities to build tribal capacity. If you're associated with a tribe, I think there are sometimes opportunities for that. They're not every year. They take some time to set up. It's not something that happens fast. So it's something that, maybe apply for funding and then it comes through a year or two later. So it really takes a lot of planning, a lot of work in advance. I don't know of any of those open right now, but they sometimes come open. And so, if you wanted to reach out to me, reach out to Gay, I think we can let you know if we find out about those opportunities. But I'll be honest that they're not every day. I don't know of any right now, but they do sometimes come up.
So Erica, this is really frustrating to hear all this, especially, and Andy doesn't know, but the Fish and Wildlife Service, which normally does a harvest monitoring tissue collection to look at the health of walruses and do other health checks with walrus or research projects with walrus. Unfortunately, they announced earlier this spring, of all springs, they are discontinuing their sampling, and they would hire local people, and it was a really great program. And they may try again in a year or two, is what we heard at the meeting. They're also hit by different financial struggles. So Erica, my best advice to answer that would be, we are on our own. We are responsible for what we see around us. We always seem to have to volunteer, and we're going to have to continue to do that. Call me, call Chuck Manadaluk at Coeric, the subsistence director. Get a hold of your AEWC rep or your any of those. Call Chuck or myself if you want to get right into NOM or ADF&G, especially if it's a land animal. That would be super duper. And what it does is eventually it helps, we will get everyone in touch, and we'll get coordinated. If you think there's an animal that has died of avian influenza, we will figure out some way to get it to the mainland, not on your dime, but we'll get it there. And then from there, the feds will probably pick it up from Nome once it comes in on Bering Air. It's okay to freeze things. If you think it's been sick, if you think it's really been sick, I mean, I don't know what to say here, Andy, because if you really think it died of avian influenza, should people be handling that? What is the current?
The official agency guidance that I've received is to not ask the public to handle samples. So that's a limitation. Exactly. But that's the official agency policy. So that's why I'd encourage folks to call and, talk with the people that if you express exactly what you see, the priority, your concern, hopefully whom you talk with is able to work with you or is able to send someone out. So I would start with reporting and getting best guidance, just to help ensure that you take care of your safety first and foremost.
Yeah, I guess I'll retract that and just say, if that's the official word, that's why we only sent 15 birds. I'll go collect whatever anybody says, but I don't ask the person to bring it into me. I'll go get it myself. But I take precautions with personal protective equipment and disinfectant and all kinds of stuff. So I guess in that case, send in photos, get it documented, because eventually that information helps these nice people help us in turn, get us some help. Hopefully there may be a chance that someone can monitor our region a little bit for birds. Who knows? But right now we're on our own. The Bering Strait region is on their own, and we will have to just do what we always do, and that is get the word out and help each other. This is about food and food safety, food security, economics. And maybe Andy, you don't know, but in Gamble, 10 pounds of Nico Calros rice is what, Gamble? $36.28 for 10 pounds of rice. So, there's a lot of pressure to make sure our foods are safe, all our seafoods and foods from the island, foods from our region, for everybody in this region. Pricing here is going through the roof. So anyway, thank you, Erica.
And maybe one more plug for reporting, how reporting can be helpful in these sort of circumstances. That, if one bird dies and the report comes in, you know, maybe someone says, well, we can't get it. But if lots of reports come in from the island, look, here's another report, here's another report, here's another report, then that's going to help make the case that something bigger might be going on and that a response is really important and really necessary. So I just want to stress that your reports are important. I've seen the records from, for instance, 2022, that data that Christina was sharing. Every one of those reports gets logged in there, even if the carcass doesn't go in. So there is a record at these agencies, how many reports are being received. Someone's looking at that and can say, well, this maybe something's going on there. So take it seriously, please. It's your choice. But if you're willing to report, I think it ultimately is going to be reviewed, and maybe we'll move the needle.
And if you think it is if you think it is a harmful algal bloom situation, we are collecting, even if there's no behavioral difference with your seafoods like upa or tunicates or any of that, the clams and things like that. Know that the Norton Sound Health Corporation Office of Environmental Health, Emma Pate, and myself are collecting specimens as well, because people are concerned about whether or not the food is getting too much of the biotoxin from a harmful algal bloom. We have very little proper monitoring offshore or near shore for this situation. And I hope that Andy and Christina, you take home like, holy cow, these people here in this Bering Strait region have quite some issues they're dealing with on our own. And so you know, that's just the way it is out here right now. But we are collecting if there's concerns about harmful algal bloom. Send in your seafood, and you can call myself or you can call Emma Pate at the Norton Sound Health Corporation Office of Environmental Health. Thank you, Erica. Holy cow, that's a long answer from us.
Thank you, Erica.
Any other questions? We're coming up on 8 o'clock. Thank you, Christina and Andy, for hanging in there. With no other questions, I'm going to make a plug for next week. I'm putting it in the chat. Next week in Nome, starting on Monday night with a reception at the museum, we have the Western Alaskan Interdisciplinary Science Conference. It is free to Bering Strait residents, but it is not free for those who come from afar, though it will be a very fun event. We urge people, if you're in Nome, come on by Northwest Campus, and I just put in the schedule. The schedule, if you're registered, there's a little bit more going on with events at the Cultural Center, at the museum, and a banquet at the VFW Hall with a no vegan option. This is no vegan option for the banquet. But anyway, come on by, and it's free for Bering Strait residents. So there are going to be a lot of people. There's topics like harmful algae and several other topics. And we've got plenary speakers that are basically all from this region. With that, next week, Strait Science is going to be, actually, we don't have Strait Science next week because we're going to be busy with the science conference at Northwest Campus, which should be a lot of fun. And then on the 17th of April, we're going to have NOAA's Ed Farley from the Alaska Fisheries Science Center in Juneau, and he's going to give us the results about the International Year of the Salmon project. Now, this project went on in the Gulf of Alaska, which seems far out from where we need to be. However, they were studying the chum salmon of Western Alaska, which we are missing at the moment in a big number. So as of last year, the chum kind of collapsed on us. Not even last year, maybe 2 years now. So he's going to be talking about what they found out in the Gulf of Alaska during the winter time, which is where our chum salmon go. He's going to have some word for us, some results for us regarding our chum salmon. So stay tuned for that. That'll be on the 17th. With that, thank you so much, Christina. And Andy, if you want to stay on for one quick second, we'll let everyone else take the night off. thank you everybody so much for coming, and really appreciate the attendance from all over the region. And it looks like we even had some people from fairly far away outside of the Bering Strait region tonight. So thank you to you guys too for tuning in. We'll see you next time. Thank you so much, everybody. Have a good night.
Thank you.
Thank you all. Thanks, Gay.
Thank you, Gay.